Okay, so today's video is going to focus on protein synthesis. We're going to do transcription and translation all today, and we're also going to talk a little bit about proteins, a little bit of review on what proteins are made of, what they do, differences between RNA, DNA, reminders of some genetic code, and then we'll get into the actual formation of the protein. Okay, so let's talk for a second about what a protein actually is. Okay, so remember, proteins are coded for by the DNA, right? And genes, and remember, these are the part of the DNA that codes for a particular polypeptide. Okay, remember, polypeptide chains are what make up our proteins. Okay, so genes are a particular portion of the DNA that's going to code for a certain polypeptide chain. Okay, and these proteins that end up that the DNA ends up coding for, these proteins are going to be responsible for our phenotype. Okay, these proteins control everything. Okay, and so they're going to have a wide variety of functions. Okay. So some reminders of some functions that proteins can have. Remember, they can act as enzymes. Okay, they can speed up chemical reactions acting as the enzyme. They can be structural in nature. Hair, silk, nails, all of those are structural proteins. They can have immune functions. Remember, our antibodies are made up of proteins. Okay? They can be, they function in movement. The cells that make up our muscles consist of proteins they're contractile proteins. Remember, the actin and the myosin that allow for the actual shortening of the muscle. Flagella are powered by proteins that are able to move. Okay? They can act as hormones. Okay, remember, we had protein and steroid-based hormones. Protein hormones were the ones that acted on the outside of the cell. Steroid hormones went all the way in. An example of a protein-based hormone would be insulin. Okay, they can also carry gases like hemoglobin in the red blood cells. Okay, they can also act as a storage site for amino acids. Okay, the example we have here is the albumin, which is what you find in an egg. Okay, acting as a storage site for amino acids. Because remember, our proteins are made up of long chains of amino acids. Remember those chains of amino acids? Those are called polypeptide chains. Okay, remember, our proteins have a primary structure. Our primary structure is our actual sequence of those amino acids, so the order that the amino acids come in. Okay, proteins then have a secondary structure. Remember, that was the alpha helix or the beta pleated sheet. Okay, and then they'll have a tertiary structure where the protein chain starts folding on itself using those hydrogen bonds, disulfide bridges. Okay, and those would all become examples of how that tertiary structure forms. And then remember, some of your proteins will have that quaternary structure when they interact with another polypeptide chain. For example, the hemoglobin has that when they interact, the multiple polypeptide chains interact together. Okay, so remember, all these proteins, these are going to determine our phenotype. And these proteins are going to come from the DNA. So we're going to be focused on how we turn that DNA into a protein. So hopefully we're going to see a lot of freshman year review right now. Okay, so remember our overview real quick of protein synthesis. Okay, remember, we've got transcription, which is the DNA to RNA, and it's going to have to occur in the nucleus, because remember, DNA can't leave the nucleus. And then that's going to be followed up by translation, which occurs in the cytoplasm with the ribosomes, and it's taking the RNA to the actual protein, or to the polypeptide chain. Okay, so those are our two basic parts of protein synthesis. Okay, so let's take a look at DNA compared to RNA, since both of these are going to be used here in this particular process. Okay, so remember, DNA. Okay, DNA is going to contain the sugar deoxyribose. Okay, while RNA will contain the ribose sugar. Okay, so that's one difference between the two. Remember, DNA is going to be double-stranded, while RNA will be single-stranded. Okay, the nitrogen bases that are in DNA, remember, DNA has adenine, thymine, cytosine, and guanine, whereas RNA has adenine, uracil, cytosine, and guanine. DNA is only going to be found in the nucleus. Okay. DNA is in the nucleus, and some organelles. Remember, mitochondria there's mitochondrial DNA. It's passed on through the mother. So some organelles will contain their own DNA. Okay, but DNA is in the nucleus and the occasional organelle. Remember, as a general rule, it's not going to leave the nucleus. That's our focus really for protein synthesis. RNA, you're going to find it in the nucleus as well as the cytoplasm. Okay, RNA is found in the ribosomes. It's in the cytoplasm attached to the amino acids. It's what take the message from the DNA. So RNA is in both the nucleus and the cytoplasm. DNA is going to be used for our RNA template as well as protein synthesis. And RNA will have multiple uses. It will act as the template during translation. It will bring the amino acids over. Like I said, it makes up part of the ribosome, so it helps attach the parts of the ribosome together. And the transfer RNA helps the transfer RNA pair with the messenger RNA. So DNA's function really is just a template for RNA and protein synthesis. RNA is going to have a wide variety of functions. So in this picture, you can see some more of the differences. Again, remember, DNA is double-stranded. RNA will only be single-stranded. Okay, and remember, there's lots of different types of RNA, but there are three main types of RNA. Remember, there's only one type of DNA. Okay, but our three main types of RNA, remember we had messenger RNA or mRNA. Okay, you have the transfer RNA, which is also your tRNA. And then you also have ribosomal RNA. And we have a few other types of RNA that we'll mention, but these are the three main ones. So the mRNA, or the messenger RNA, this is the RNA that takes the information from the DNA out to the cytoplasm. Okay, remember, this is what's going to determine our amino acid sequence for the protein, because what amino acids come over and bind together to form the polypeptide chain is based on what this messenger RNA says. Okay, remember your transfer RNA, this is the one that brings the amino acid to the ribosome and the messenger RNA during the process of translation. Okay, and then the ribosomal RNA is part of the ribosomes. It acts as a connection, kind of, between the messenger RNA and the transfer RNA. Okay, so all of these have a big role in protein synthesis. Again, your messenger RNA is taking your information from your DNA to the cytoplasm. It's going to determine the sequence of the amino acids for the protein. The transfer RNA is going to bring the correct amino acid to the ribosome during translation, and the ribosomal RNA is going to connect the messenger RNA and the transfer RNA together. We also have a few other kinds of RNA that will get mentioned a little bit later. We'll talk more about them. Okay, we have the siRNA, which this is found on spliceosomes. Spliceosomes are what cut out the introns. Okay, if you remember, introns are the portion of the DNA that don't code for anything, where you'll have a long chunk of DNA that'll be made up of introns and exons. Introns are the non-coding or junk DNA. Your exons are the DNA that actually are used to and are actually translated into proteins. You also have your SRP. Oh, I can't write. That's P. SRP RNA. Okay, and this is part of a signal recognition particle. And that signal recognition particle is going to be, it's going to help the ribosome kind of thread the polypeptide that's being formed from the ribosome. If it's going to be exported out of the cell, it's going to need to be processed further by the endoplasmic reticulum. And these will help get that polypeptide chain into the endoplasmic reticulum so that that can happen. There are going to be some differences between protein synthesis in a eukaryotic cell versus a prokaryotic cell. So in a prokaryotic cell, there are no introns. Okay, so all of the DNA in a prokaryotic cell is coding DNA. There's none of this junk DNA that needs to be cut out and removed. And so in prokaryotes also, the messenger RNA does not have to be processed. And we'll talk about this more in detail in the eukaryotic process. But in the eukaryotic cells, it needs to be, some modifications need to be made, the introns need to be removed. And so in prokaryotic messenger RNA, that does not have to happen. Okay. Also in prokaryotes, transcription and translation can happen at the same time. Okay. They can be simultaneous. And that's going to be true for prokaryotes only. Okay, so again, your transcription and translation can be simultaneous in your prokaryotic cell. Oh, I just ran out of room. So you can see in your pictures here, you've got your eukaryotic cell here on the right, okay, and you see you have this pre-RNA. Okay, that's because that RNA has to be processed before it's sent out for translation. Here in the prokaryotic cell, it's just a little bit simpler. There's no introns, there's no processing happening, and transcription and translation can happen at the same time, okay, mainly because there is no nucleus segregating the two. Let's do a little genetic code review. Remember, genetic code is the genetic code is redundant. Okay, remember, you're going to see some of the same. There's more than one code for a lot of the amino acids that are out there. Okay, remember your codon chart when you would look at these, and you would see multiple codes for the same amino acids. Okay, remember your amino acids are coded for in groups of threes. Okay, they're coded for in the groups of threes that are called codons. Okay, and when we use those groups of three to read our chart, we can determine which amino acids we have. There are codes for both stop as well as start. Okay, start is most of the time is our methionine. You can see that right there. You can see you've got your 3 stop codons there as well. And remember, your genetic code is also considered universal. The majority of organisms use the exact same code. So what I mean is that how we see here, CUU codes for leucine. That would code for leucine in a plant, in a wheat plant, in a palm tree, in a horse, in a cricket, in a person, in a mushroom. Okay, CUU, that's universal. That's always going to code for leucine. And that is, there are exceptions to that, but as a general rule, this code is universal. This is one of the ways that we can produce, for instance, synthetic insulin. Prokaryotes can read eukaryotic genes because of this universal genetic code. Okay, so let's take a look at how these proteins are going to actually be made. Let's start with our transcription process. So remember, transcription is the process basically of taking that DNA into a messenger RNA template. And we focused, our focus from this year, freshman year mostly, was getting that concept of DNA to RNA, where it happened, and we're going to take that a little bit further. We're going to look a little bit more detail as to how that's actually going to happen. Okay, it's a multi-step process. Okay, it's got an initiation portion, an elongation portion, and then a termination of it. And then that RNA, again, is going to have to be processed so that it's ready to be translated. Okay, so let's zoom in here and narrow our focus at first to the initiation process. How are we going to start this? Okay, so we're going to start this by RNA polymerase is going to bind to a promoter region. Okay, so the RNA polymerase enzyme is going to bind to the promoter region. And you'll see the promoter region is before the actual gene really officially starts. Okay, and so the promoter region is going to determine which side of the gene is going to be transcribed. And the RNA polymerase will attach there. In a prokaryotic cell, it will attach directly. Okay, so that'll be a direct attachment. In a eukaryotic cell, this is going to require transcription factors. Transcription factors are going to be a series of proteins that are going to help basically enable and facilitate the binding of this RNA polymerase. And again, these are only in eukaryotes. So in eukaryotic cell, these transcription factors facilitate or aid in